Is type O positive blood actually African? DNA. Just exposed. What's been hidden for centuries? What if the most important thing about your blood isn't your blood type, but the journey it took to reach you? If you have type O positive blood, you're carrying a genetic signature that's older than your surname, older than your nation, and older than every civilization that has ever existed. Most people see O+ on a medical form and think it's just a label, a letter, a symbol, nothing more. But when geneticists began extracting ancient DNA from Ice Age skeletons and tracing humanity's earliest migrations, they discovered something extraordinary. Those two simple characters aren't just medical information, they're a record of survival written into your DNA. A record left behind by people who crossed frozen land bridges, survived deadly pandemics, endured ice ages, and reached places no human had ever set foot before. Today, type O positive is the world's most common blood type, carried by nearly 38% of humanity. For decades, scientists believed that common meant ordinary. They couldn't have been more wrong. Modern DNA research has revealed that type O positive may be the oldest surviving bloodline in human history. Its origins reach far deeper than anyone imagined, and the path it took across the world rewrites much of what we thought we knew about our ancestors. By the end of this documentary, you'll understand why those two small characters on your medical record may tell one of the oldest stories ever written, and why you'll never look at your blood type the same way again. The story of blood typing begins in 1901 in a laboratory in Vienna. An Austrian physician named Karl Landsteiner was trying to solve a deadly problem. Blood transfusions kept killing people. Patients who received donated blood often died within hours, their bodies seizing as their immune systems attacked the foreign fluid. No one understood why. Landsteiner noticed something under his microscope that others had missed. When he mixed blood samples from different people, some combinations clumped into thick, sticky masses. Others stayed smooth. The clumping was the immune system rejecting something it recognized as alien. He had just discovered that human blood was not one thing. It was several. He identified 4 groups, A, B, and O. Landsteiner received the Nobel Prize for it in 1930, but the discovery came with an assumption baked in from the start. Scientists looked at blood type distribution and drew what seemed like an obvious conclusion. Type A appeared everywhere. It seemed dominant, foundational, original. For decades, that assumption went unchallenged. Blood typing became routine in hospitals and militaries worldwide. But the deeper question of which type came first was left largely alone. It would take the sequencing of DNA itself to prove that the original assumption was not just incomplete, it was completely reversed. When geneticists began sequencing the gene responsible for blood type, they were following a technical question. The blood group system is controlled by a single gene on chromosome 9. That gene instructs an enzyme that decides whether sugar molecules called antigens get attached to red blood cells. Type A adds one sugar, type B adds another, type AB adds both, type O adds neither. The enzyme in type O carriers is broken. A single missing base pair renders it inactive. Simple enough. But when researchers compared that broken gene across thousands of individuals from dozens of populations, the timeline stopped them cold. Molecular clocks showed that the type O variants are extraordinarily ancient. The 2 most common forms, O1 and O2, diverged well over 1 million years ago according to molecular clock estimates. Homo sapiens as a species is only around 300,000 years old. Type O predates us entirely. It was already present in our ancestral lineage before modern humans existed. Type O is not a simplified version of A or B. It is the original template. A and B are the mutations. They arose later when single nucleotide changes added new enzymatic functions on top of a foundation that type O carriers have never lost. The blood that seemed most ordinary turned out to be the most ancient thing inside you. Once researchers established that type O was the ancestral form, the next question became geographical. Where did it concentrate most heavily, and what did that reveal about the path it traveled through history? The global distribution map produced an answer that surprised almost everyone outside the field. The deepest and most genetically stable reservoirs of type O positive on Earth trace back to Asia, not uniformly. The continent is vast and its populations are diverse, but when researchers isolated the specific DNA variants inside the type O gene, the signatures pointed repeatedly to East and Southeast Asia as the origin point of the most ancient and widespread O lineages. Across Southeast Asia, type O appears in roughly 40 to 45% of tested populations, with consistent prevalence from the Philippines through Vietnam and into southern China. The particular mutations that produce type O in these populations, especially variants labeled O1 and O2, carry surrounding DNA markers that function like a return address. Small variations in the flanking genetic sequence allowed researchers to trace those lineages backward through time. Every trail led into the deep past of the Asian continent. Ancient DNA extracted from remains across East Asia showed that type O was already deeply established in these populations tens of thousands of years ago. The Asian fingerprint on type O positive was not recent. It was written into the genome before recorded history began. Asia did not keep its blood to itself. Between 15,000 and 20,000 years ago, during the last ice age, sea levels dropped dramatically as glaciers locked water away from coastlines. In the far north, a land bridge emerged between what is now Siberia and Alaska. Researchers call it Beringia. At its widest, it stretched hundreds of miles, a broad, cold plain connecting two worlds that had never been connected before. Small groups of ancient Asians followed game animals across it and walked into a continent no human had ever entered. They carried almost nothing except what was written inside their cells, and what was written there was overwhelmingly type O. Population geneticists running simulations using dental remains, Y chromosome diversity, and mitochondrial lineages consistently show that the founding groups who crossed Beringia carried type O at frequencies well above average for the broader Asian populations they came from. Once inside the Americas, isolated and exposed to new disease environments, those frequencies only climbed. Genetic drift and selection pressure pushed type O toward dominance. In some indigenous communities of the Amazon and the Andes, it reached near total exclusivity. Every individual tested was type O. No A, no B. Nowhere else on Earth does a single blood type dominate so completely. The Great Migration was not one journey, it was many, and type O rode every single one. Type O did not survive by accident. It survived because in the specific environments where it became more concentrated, it offered something other blood types could not consistently match: a slightly better chance of living long enough to have children. That edge was never absolute. Type O is not a shield, but across hundreds of thousands of years, even a marginal survival advantage compounds into something measurable. The clearest example is malaria. The parasite responsible for the most lethal form, Plasmodium falciparum, uses sticky proteins to clump red blood cells inside capillaries, cutting off oxygen to vital organs. That clumping happens less efficiently with type O red blood cells. The surface chemistry reduces the parasite's grip. In regions where malaria shaped human genomes for millennia, type O frequencies remained high. Certain gut viruses also bind less effectively to type O cells. In tropical environments flush with waterborne pathogens, that molecular difference may have translated into lower rates of severe illness during early childhood, the period when disease kills most efficiently. Populations that survived those environments generation after generation were disproportionately type O, not because type O was chosen, but because everyone else died slightly faster. That is how evolution works, quietly, ruthlessly, and over an almost unimaginable stretch of time. One thing before the final piece: everything I just showed you about type O positive blood is traceable inside your own DNA results. Type O positive is no exception. The same ancient chemistry that helped its carriers outlast plagues and resist parasites also left them exposed in ways that took centuries of research to document. Start with the stomach. Type O carriers have a measurably higher lifetime risk of peptic ulcers. The mechanism involves Helicobacter pylori, one of the most common chronic infections worldwide. H. pylori thrives more aggressively in the stomach environments of type O individuals, gaining a stronger foothold due to the same surface chemistry that resists certain parasites. In East Asian populations, where type O frequencies are high and H. pylori exposure has historically been widespread, researchers have documented elevated rates of gastric cancer tied directly to this interaction. Then there is clotting. Type O carriers have lower levels of von Willebrand factor, a protein central to blood clotting. This means slightly thinner blood and reduced risk of dangerous clots, but it also means heavier bleeding when injured and slower trauma recovery. In ancient environments, a severe wound could be fatal faster for a type O carrier. There is also the Bombay phenotype, a rare anomaly found most commonly in South Asia, where individuals appear type O on standard tests but are genetically distinct at a deeper level and cannot safely receive standard type O blood. Genetics never offers anything for free. For most of the 20th century, everything scientists knew about ancient blood types came from inference. They mapped living populations, ran models, and worked backward. It was educated guesswork. Then ancient DNA technology arrived and changed everything. In the past 15 years, researchers have pulled readable genetic material from remains thousands of years old, bones in permafrost, teeth in collapsed caves, and skeletal fragments from Neolithic graves. The DNA was degraded and fragmented, but with the right tools, it became legible. Researchers found type O dominance in East Asian remains dating back tens of thousands of years, well before any recorded civilization, confirming its long-standing presence long before the Beringian migration. They found that ancestors of modern Native Americans carried type O at even higher frequencies than their living descendants, suggesting post-contact disease events shifted surviving gene pools. They examined remains along early Silk Road routes and found type O overrepresented among the ancient populations who first connected East Asia to Central Asia. Those traders carried their blood type along the same paths they carried silk and spices. Populations descended from those networks still carry the genetic echo today. The DNA of people dead for millennia confirmed what the DNA of the living had only suggested. The Beringian crossing is the migration story most people know, but there is another journey in the type O positive record just as astonishing. Around 5,000 years ago, seafarers from what is now Taiwan set out across the Pacific. They had no compasses, no written maps. They had outrigger canoes, deep knowledge of ocean currents and stars, and the courage to sail toward nothing visible on the horizon. Over the following millennia, their descendants spread across the entire Pacific. They reached the Philippines, Indonesia, and Melanesia. They settled Hawaii, Easter Island, and New Zealand. They may have contacted the South American coast long before any European ship came close. Researchers call this the Austronesian migration, one of the most extraordinary feats of navigation in human history. And almost everywhere these seafarers landed, they left a genetic signature, elevated type O frequencies. The Polynesian populations descended from this migration carry some of the highest type O concentrations on Earth, alongside indigenous American communities, two groups separated by the world's largest ocean, all carrying the same ancient Asian blood inheritance. The ocean did not divide it. It carried it forward intact. Blood type has never been just about what sits on red blood cells. The ABO gene sits inside a broader cluster that also regulates immune response to pathogens. For type O carriers, that neighborhood matters. The gene FUT2 sits near the ABO gene, and the two are not independent. FUT2 controls whether blood group antigens are secreted into bodily fluids, saliva, mucus, and the digestive tract lining. Individuals with an active FUT2, called secretors, release these antigens into their fluids. Those antigens act as decoys. Norovirus and rotavirus strains bind to gut sugars as their entry point. When antigens are present in the mucus layer, the virus binds to them instead of the cell wall. The infection is blocked before it begins. In indigenous populations across the Americas, where type O frequencies approach 100%, researchers found a distinct haplotype linking type O with elevated FUT2 expression. Communities carrying this combination reported significantly lower rates of severe gastrointestinal illness, even with poor sanitation. Lab studies confirmed the resistance. Epidemiological surveys matched the benchmark. What began as a footnote about sugar molecules turned out to be a window into how ancient populations survived environments that should have killed far more of them. Modern medicine is still decoding what blood type truly means. New studies connecting ABO genetics to disease risk, immune response, and population history appear every few months. Type O positive is not just the most common blood type on Earth. It is one of the most information-dense biological signatures the human body carries. Researchers have linked type O to slightly reduced cardiovascular risks tied to lower von Willebrand factor levels. They have found distinct mosquito attraction patterns in type O carriers, relevant in regions where mosquito-borne disease remains a leading killer. Early data from COVID-19 studies suggested type O carriers experienced modestly lower rates of severe infection. None of this makes type O superior. It makes it a record. Every association points back to the same story. Ancient populations in specific disease environments survived at slightly different rates, depending on what their blood carried. Those differences accumulated across thousands of generations into the distribution we see today. When you look at those two characters on your medical chart, you are not reading a category. You are reading the outcome of an unimaginably long series of survival events, stretching back through ice age migrations, tropical plagues, ocean crossings, and over 1 million years of molecular history. Your blood has been keeping score the entire time. There is a quiet truth sitting inside every type O positive carrier alive today. Your blood did not begin with you. It did not begin with your parents or grandparents or any ancestor whose name you know. It began in ancient Asia, in populations that crossed frozen land bridges and open oceans, and that outlasted plagues and ice ages and bottlenecks that should have erased them. It persisted not because anyone chose it, but because the people who carried it survived just slightly better, generation after generation, across an almost unimaginable stretch of time. That letter on your medical chart is not administrative. It is a timestamp. It marks you as the living descendant of some of the most resilient travelers in human history. Modern science is still decoding exactly what that means, New studies appear every few months. Ancient DNA keeps pushing the story deeper into prehistory, but the broad outline is already clear. Type O positive is the ancestral blueprint, the oldest widespread blood signature the human body carries, and it rode the first great migrations out of Asia to become the most common blood type on Earth. You did not inherit ordinariness, you inherited endurance. The DNA inside your red blood cells is one of the oldest artifacts you will ever carry, and the story it tells has been waiting tens of thousands of years for someone to finally listen. Most people look at their DNA percentages and stop there, missing the real story. If you enjoyed this documentary and want to discover more hidden stories behind human history, genetics, and ancient civilizations, subscribe to the channel and turn on notifications. Every week, we uncover the mysteries that history and science are still trying to explain. Thanks for watching, and I'll see you in the next documentary.